Um, I know. I don't know. So I just want to remind you of your liver list. Liver list? Sound vaguely familiar? Why would you want to do it? 5% on your final, which is free. Yes, which is the only extra credit that's available on your final. So it behooves you to make sure that you turn it in and spend more than 10 minutes on it. Because, like, literally, you could search the PDFs of all of your notes for the word liver. Duh. It's not that hard. Okay. And yeah. Yeah, you have to have a body system on it. Huh? Well, then put a couple of systems. Yeah. I mean, as long as it's obvious that you know what you're talking about, I'm not going to ding you for leaving off of what? Yeah. Uh-huh. Yeah. And what happens if you don't bring it when you take your final? You do not get credit. I am not taking it later. I am going to London the day after finals. I ain't taking any late papers. I won't be busy. Right? I won't be on a plane. Okay. All right, so we are going to do blood. We will almost finish it today. We'll get through all of the hard stuff. And then we just have respiratory and urinary. Both of those are only about a lecture and a half, so we're on track with finishing them. And they're not even outrageously hard as long as you make sure that you understand the little relationships before you move on to the next ones. Basically what they're going to do is, you know, carrying oxygen and carbon dioxide around, right? They're going to regulate the pH of the bloodstream. And then, of course, your urinary system is going to regulate both blood volume and also waste products in the bloodstream, and then some other stuff that we'll talk about. But it's not outrageously difficult. I do not think that the fourth test is as hard as the third test, but I do think that what happens is everybody gets dumb at this time of the year, so don't let it happen, right? Your brain's like, I got 20% less processing power than I did at the beginning of the semester. Okay, all right, so, but somebody asked me in the last class, is it the easiest test? No, it's not the easiest test. The first test was the easiest test. If you go back and look at it, you'd be like, who even got that wrong? Who the hell was that? I got cellular respiration wrong. How'd that happen? Okay, so we are going to do blood. Here's how I do blood and phys. I'm not doing blood globally, and I don't do immunology, not because it's not important, but because I know that you will get it intimately and deeply when you take micro. You do immunology with a lot of detail. Are you taking micro at the same time? She's taking all 3 classes, anatomy, physiology, and micro at the same time. Crazy person. She hasn't smiled like in 6 weeks, though, honestly. She's like, I hate all of you and never want to talk about another biological principle. Okay. Okay, so immunology is super important, very important physiological process, but I know that you're going to do it in detail. So I am going to talk primarily today about red blood cell function and clotting, which is related to platelet function today. Before we get into that, let me just acknowledge the fact that plasma exists and is important, right? So plasma is generally described as a straw-colored liquid. It's about 90% water, and its primary function is to carry around water-soluble stuff from point A to point B, yeah? So what do you carry around in plasma? Well, anything water-soluble that you're trying to get from point A to point B, like your nutrients, the water-soluble ones, are carried in your plasma, meaning not inside your cells, but in your plasma. Your electrolytes, those are all water-soluble. They travel in the plasma, right? Your respiratory gases. Now, the vast majority of your oxygen travels inside your red blood cells on what protein? Hemoglobin, of course, but some of it travels in the plasma. And carbon dioxide is the opposite. Carbon dioxide, most of it travels in your plasma, but a little bit of it travels on your hemoglobin. Okay. And waste products, any water-soluble waste products like nitrogenous waste products, urea, uric acid, ammonia, those travel in the plasma as well. And then last stuff that travels in the plasma is clotting proteins we're going to talk about today. Most of them are made by the liver. Ding, ding, ding. And then the other proteins that are really abundant in the blood plasma are albumin. Albumin is one of the most abundant proteins in your body. I think albumin and collagen are the two most abundant proteins in your body. But albumin is produced by the liver, and it does a couple of things. First thing is, it is a really common escort protein for lipid-soluble substances. Remember, lipid-soluble substances have basically ADHD, right? And they need somebody to hold their hand all the time. So this is a really common escort protein for lipid-soluble substances. The other function is this. Do you guys remember how we talked about at the arterial end of a systemic capillary bed, you're going to be high pressure, so you're going to lose some fluids via filtration. Yes? Okay. And at the venous end, you are going to suck most of it back up by osmosis. Yes? Okay. So what is the major osmotic draw on that side? It's that there's a whole bunch of albumin that is trapped in the blood plasma. And since it's trapped in there, it's basically sucking the fluid. So that means that albumin is helping you to maintain your blood volume, right? Because if you've got a whole bunch of albumin, it's going to pull most of the water back up that leaked out by filtration, okay? So if you have abnormal albumin levels, it can impact your blood pressure. Okay, so sodium ions will also help. And the other thing that albumin does is it actually has a little bit of buffering capacity to help to regulate the pH of the bloodstream. Okay, the other proteins are alpha, beta, and gamma globulin. So let's talk about gamma globulin first. Have you guys ever heard of a gamma globulin shot? No? Okay, so gamma globulins are antibodies. They're produced by lymphocytes during the immune response. Okay, so if I get exposed to measles, yes, I produce antibodies that are specific to measles, yeah, part of the immune response, yes. So those are traveling in the bloodstream and they're protective, and of course they're the smart part of your immune system that builds with exposure or builds with vaccination. So the reason that I thought you guys may have heard of gamma globulin shot, and far more people have heard of them here than in Texas, is because quite often, and a big part of Utah culture, people are going to go on missions to foreign lands, right? And if I am going on a mission to Central Africa, being a North American girl, it's unlikely that I've ever been exposed to dengue fever, yeah? So how the hell do I keep myself from getting taken down by dengue fever during that first initial exposure? Well, I shoot myself up with the antibodies that are specific to the pathogens that I might encounter in that part of the country. Does that make sense? So I shoot myself up with those antibodies, and what it does is provides a little window of protection for me to make my own antibodies. Yes? Okay, so there would be a different one for Central Africa, and there would be a different one for East Asia, and a different one from South Asia, and a different one for the southern part of South America, right? Now, it's just a little protective mechanism. They call them peanut butter shots because apparently they're really, really viscous and super painful when they inject them. Has anybody ever had one in here? No? They also do it for the military, right? So is that the same as a vaccination? No, but you're probably going to get vaccinated too. What a vaccination is instead of injecting you with the antibodies, they inject you with the antigens so that you will make the antibodies. Yes? Okay, so they're giving you, so you could actually do both, right? If you wanted a person to sort of have that protection, right? You could actually do both. Does that make sense? So that's gamma globulins, and those are made by lymphocytes primarily during your immune response with exposure, but you can also take them, they can make them in the lab for you too. Okay, alpha and beta globulins, those are primarily, again, escort proteins for either lipid-soluble stuff or also some iron-based stuff. And they named them globulins literally because IN means protein, and they thought they were glob-shaped. So somebody called them globulins, which I think is kind of cute. You imagine with like 17 PhDs, let's call them a globulin. Okay, so they've got clotting proteins, and then there's other proteins in there. Some of your hormones, or of course the water-soluble hormones are in there. All right, so now let's get into blood specifically. So just a little general information before we get to red blood cells. What is blood like? It is thicker than water. Yes, it's about 5 times as viscous as water. Yes. It's about the texture of like whole milk to half and half between those. And of course, it can get different if you're like really, really hydrated or dehydrated or something. It's pretty sticky, primarily because it's got all those sugars and proteins in the blood plasma. It's usually a little hotter on average than your whole body temperature because it's going through really highly metabolizing organs and then out and really highly metabolizing organs. Its pH is really tightly constrained, 7.35 to 7.45. Go very far from that either direction and you will start to change the shape of your proteins. So we will learn in the next two sets of notes that what primarily constrains the pH is a combination of respiratory system and urinary system, really, really controlling the pH of the blood. And it is isotonic to about 0.9% saline, right? So if, for instance, you get dehydrated and you don't have other problems, they will add normal saline back, right? Which is about 0.9%. It makes up about 8% of your total body weight. So that's that last 5 pounds you were trying to lose. Don't even try. You need that blood, right? And then males typically, because larger body size, they have more blood on average than females, okay? This is just because of larger body size. Males usually have between 5 and 6 liters, and females have between 4 and 5. But we will see that the blood is also different in males and females too. Okay, so basic functions of blood. You know this stuff. It's carrying stuff from one place to the other. It's carrying waste primarily to your kidneys, your liver, and your skin. It's carrying oxygen from your lungs to your cells so that you can use it for what? Cellular respiration. It's carrying oxygen, carrying carbon dioxide from your cells to your lungs that you made from cellular respiration, nutrients, GI tracts to cells, all of that kind of stuff. So primarily it is doing a bunch of transport, but in addition, it is also doing some regulation. It's regulating pH because it's got buffers in it. It's regulating ion composition. For instance, if I need a whole bunch of calcium in the brain, but I have it in the bones, I just carry it from one place to the other. And it is the only fluid we will see that can regulate fluid volume because it's the only fluid you have that has the ability to clot. Okay? So, and then it protects you from fluid loss and it protects you from microbes and toxins because it's got white blood cells and antibodies in it. And it also is working like a swamp cooler to stabilize your body temperature because what it's doing is it's going through hot organs and bringing them close to cool skin. Hot organs, does that make sense to you guys? So it's basically got swamp cooler activity just by the circulatory mechanism. And then in addition, it also helps to regulate body temperature by forming and breaking hydrogen bonds, and that's a way of regulating body temperature. Okay, so that's the simple stuff that you probably have known before. Now let's get into red blood cells a little bit. Red blood cell physiology. Okay, so your red blood cells, if we look at whole blood first, if you spin whole blood down in a centrifuge, it will separate it by density. Yes? Anybody have any idea why these are so heavy and dense, red blood cells? What's in them? Because they have something even heavier. Yeah, they got iron in them. So they tend to fall to the bottom. And notice that that's like 45-ish%, but that varies. It varies. It's different between males and females too. And then the buffy coat is just the white blood cells and platelets, and then notice that the rest of it is blood plasma. Now, these proportions vary a little bit between the sexes, and they also vary between whether you're hydrated or not very hydrated. But that is the whole blood. And so if we start looking at red blood cells specifically, let's talk about where you make them. You should know. Where do you make red blood cells? Where are they produced in the body? Be more specific. Red bone marrow specifically. All right, so you're going to make your red blood cells in the red bone marrow. Note to self, you've probably heard this word before, but maybe not. The process of blood cell production globally is called hematopoiesis, but a subset of that is called erythropoiesis. And erythropoiesis is specifically the production of red blood cells. Does that make sense? So what we're going to do now is talk about the process and the needs for erythropoiesis. Everybody good? Okay, so what do you need in order to do erythropoiesis? Well, you're going to need all of the precursors to making an entire red blood cell. So of course, does it make sense to you that you are actually going to need iron in order to do this? Yes? Okay, so let's talk about iron a little bit. Do you guys remember ever seeing a picture of hemoglobin before? It's a quaternary protein. It's got 4 different amino acid chains, 2 alpha subunits, 2 beta subunits. And at the center of each one of them is a heme group, and at the center of the heme group is an iron atom. Is that good? Okay, so On each heme group is where oxygen gas would ride. Yes? So imagine that this is a little oxygen ferry, not flap, flap, but toot, toot, that kind of ferry that carries things around. Ready? Okay, so you know, but tell me anyway. How many oxygen gas molecules can you carry per hemoglobin? 4. Yeah, got it. Okay, keep that in mind because we'll talk about how you carry carbon dioxide as well. So this is a protein. And in order to make it, you are not only going to need amino acids in your diet, and of course you're going to need ATP for protein synthesis, but in addition, you're going to have iron. So where do you get iron in your diet? How do you lose it? How do you replace it? Where do you store it if you have extra? Okay, sources of dietary iron first. The primary sources of dietary iron are meat, liver, shellfish, egg yolks, beans, nuts, cereal, leafy green vegetables. Now, notice that there are ways that you can get adequate iron in the absence of eating animal products. If you are vegan, you're going to have to pay a little more attention to your sources of iron to make sure that you get enough of them. So how do you lose iron? Well, most forms of iron, but not all, are water soluble, so you lose them when you lose fluids. So you lose them via urine, skin, sweat. And of course, women who menstruate are going to lose iron via menstrual flow because when you menstruate, you know this, but I never make any assumptions about what people know about the reproductive tract, reproductive systems in either Texas or Utah because they are both abstinence-only sex education states. So if I'm insulting your intelligence, it ain't about you. It's about him, the other dude, or her. Honestly, women are way worse with the reproductive system than males. They don't even know where their parts are. They put the clitoris all up in the urethra. I'm like, no, it ain't there. And I'm like, we could improve your quality of life so dramatically. Okay. So, what? That should be my platform. I got run out of the state again. Okay. So menstrual fluid is not just fluid; it's actually tissue. And it's really, really vascular tissue. So when a woman menstruates, she is going to lose tissue with lots and lots of blood vessels in it. Does that make sense? So what tends to happen with women who are menstruating, is if you look at their iron content in their bloodstream before they menstruate, it will be relatively high, and then it'll drop, and then it'll go back up, and then it'll drop, right? Okay. With men, it does not tend to be very cyclical. Their iron content in their blood tends to be relatively constant. That makes sense? So you do lose iron via that mechanism. But you also have mechanisms of changing how much iron you absorb and excrete. So you absorb iron primarily, of course, via your digestive tract. But if your iron level is low, you'll actually absorb more. And what do you predict about your excretion? If your iron level is low, you'll excrete less into your urine, right? So there's a little bit of homeostatic control that you can do with your absorption and secretion and excretion of iron. Does that make sense to you guys? Okay, so if you ever have extra, and this is important to understand, what you generally do is you store it in your liver. That's where your extra storage of damn near everything goes. You store so many things in your liver. Go ahead.
Too much iron, what could that cause?
It causes a disorder called hemochromatosis, and males are far more likely to have it than females. And hemochromatosis causes problem with carrying things around in the bloodstream broadly. Okay, and any idea how you would treat a male who had way too much iron in his bloodstream? No. What might you do? I mean, you can't make him menstruate, but what can you do? He can do therapeutic phlebotomy on a regular basis, usually about a monthly basis, sometimes every 3 weeks, depending on how high his iron level is. He just basically gives blood, right? And interestingly, it decreases the likelihood of certain blood disorders that women don't get and males do. Like not just that one, but other blood disorders. Apparently having cyclical levels of iron tends to be beneficial in some ways. Go ahead.
Can they still use that blood?
Usually they can't use it for whole blood, but a lot of times they can use it for plasma or platelets. That makes sense? His whole blood is a little off, but plasma and platelets is fine. Is that okay? Okay, so if you have excess iron, you store it in your liver. And of course, that's why if you are iron deficient, sometimes you eat liver of other critters, not generally of humans, although I've seen Silence of the Lambs too. So I, being the little redneck kid that I was in Texas, right, I was relatively anemic. when I was a kid, and it tended to be pretty iron deficient most of the time. And what did my mom do? My mom, my good Southern mother, fried up some chicken livers and stuck little toothpicks in them, and they were on the bottom shelf of the fridge, and I'd just toddle on in there and eat myself a chicken liver. I also really liked spinach back then. So of course, that probably dumped boatloads of cholesterol into my system, because that's also where you store cholesterol, but whatever. I lived. Okay, so excess iron is stored in your liver. Okay, so what else do you need? Of course, you need proteins to do protein synthesis, but you also need some things to do cell division. These two are primarily related to cell division. Okay, so before you do cell division, you need to do DNA replication. Yes? Everybody with me? I know you hate it when I talk about 1610 stuff, but does it sound vaguely familiar that you need to replicate your DNA before you divide your cells? Okay, so you make 2 to 3 million red blood cells per second. So you got a boatload of work to do right here. And so you are going to need to do a boatload of DNA synthesis. Specifically, you need folic acid for making thymine. Now, you need it for doing DNA synthesis no matter what cells, but you're doing a bunch of it in your red blood cells, and you really never stop. So note, sources of dietary folic acid: green plants, liver, yeast, and citrus are your primary sources. Okay, so I'm just going to go on a little aside here, because if this isn't something you've heard before, it's useful to hear. So at one point in a woman's life, it's a big old cell division party. What point is that? Pregnancy. So if she is pregnant, yes, big old cell division party. One of the things that we figured out pretty late in the game, honestly, they figured it out, I think, in the 1970s, and then educated really dramatically about it in the 1980s, was that an absence of adequate levels of folic acid during pregnancy causes very, very preventable nervous system development problems in the fetus, like neural tube defects like spina bifida. And so a woman, in order to get through a healthy pregnancy and to have the nervous system develop normally, you need relatively high levels of folic acid the entire pregnancy, starting from about 2 weeks post-implantation. Now, those of us who have been moms, did we know we were pregnant 2 weeks post-implantation? I sure as hell didn't. I did not know. So the reason that this needed a big education campaign is because there were a whole bunch of preventable neural tube defects in spina bifida. But if a woman knows that she could possibly become pregnant and would not choose to terminate a pregnancy, what should she probably be making sure is in her diet? Folic acid, yes. And if you're not eating beans, nuts, cereal, and leafy green vegetables, you can pop a women's multivitamin and it'll have adequate levels of folic acid. And you might not need a damn thing in there but the folic acid, but you only get one shot to put together a nervous system, right? And they have reduced this, the predisposition or the frequency of neural tube defects by that education campaign. Okay, so that's not directly related to this, but I had to say it. Okay, vitamin B12, by the way, primarily in animal products. I say only, but I should have put an asterisk there, because it's in high quantity in animal products, and it's in relatively low quantity, but if you are eating the right plant and fungal products, you can still get it. So it's also needed for DNA synthesis, so these two are buddies right here. Okay, and then another hormone, because I know you want one. Okay, EPO is a hormone called erythropoietin. What is it? It's a peptide, exactly. It is a hormone that comes from the kidney and targets the bone marrow to cause erythropoiesis. So you can't tell me why it would be released. I'll give you two choices. It would be released from the kidneys in response to low levels of oxygen or high levels of oxygen. Low levels of oxygen, yes. That's the trigger, hypoxia, as detected in the kidney. So if hypoxia is detected in the kidney, it releases EPO, erythropoiesis, and causes the bone marrow to do erythropoiesis. Is that good? Erythropoietin. So let's talk. Of course, you have to have iron in order to do it, but assuming that you have iron. All right, so this erythropoiesis produces 2 to 3 million red blood cells per second, as I mentioned. There is a lot of work that is going on there, and it's not constant. That's an average of your level. So let's just talk conceptually about the reasons that your kidneys could be encountering hypoxia. Like, why would your kidneys be encountering blood that is low oxygen? There's a whole bunch of different reasons. Go ahead. Oh, yeah, high altitude will totally do it. So decreased availability of oxygen in the atmosphere, right, means that is not going to be as much oxygen in your bloodstream. This is what I encountered, of course, when I moved from Texas to here. It's high altitude, right? Oh, you moved from freaking Brazil to here. Oh, my gosh. But did you move from mountainous Brazil or?
Oh, sea level.
Oh, beach Brazil. Oh, my gosh. You must have been like, what the heck? If I, for instance, was from sea level and he's from sea level and we got here, what that means is there was abundant oxygen in the atmosphere. So we didn't have very many oxygen ferries, yes, because we didn't need that many. So what happens is we get here and we start encountering hypoxia, right? All the damn time, an error signal that says you do not have enough oxygen, you don't have enough oxygen. And then once our bodies trusted us that we were actually going to stay, then the kidney starts releasing EPO. Will it do it if you fly somewhere for a day? No, because the error signal is not consistent for long enough to do something so expensive. But after we stayed for a little while, it causes it to release. So decreased availability of oxygen, COPD, which is chronic obstructive pulmonary disease, someone who can't get enough air in will also release it. This one is interesting. If you have a decrease in red blood cells, like if you hemorrhaged or if you gave blood, yes, or if you had destruction of your cells with like a mismatched blood transfusion or something like that. Does that make sense? So all of those would cause EPO release as well. I did not know this the first time I gave blood when I was 18. I was an English lit major, so forgive me. I gave blood and then I went and worked out. What the what? I need some education, didn't I? And it took me 72 hours to recover from it. Yeah, oh my God, it was so terrible. Yeah? You probably got a little more blood than I do, too. Okay. You shouldn't do that to yourself. Okay, increased demand for oxygen. So if you start exercising, but not once, not January 2nd, like we all do, right? I'm going to be amazingly hot this year. No, that's a lot of work. But if you do it regularly, your skeletal muscles are using up more of the oxygen, right, regularly, so you continue to give that error message to your kidneys, and then your kidneys will say, oh, okay, I'm going to get you some more red blood cells. But it doesn't happen unless you do it regularly. Go ahead.
So what are the physical attributes or whatever you want to say for the altitude change? The difference? What happens
In me?
Anybody, besides feeling like you're going to die walking.
Oh, yeah, that you're feeling like you're going to die. You're completely exhausted. You have absolutely no energy. Usually your skin is relatively pale compared to how it usually is. Shortness of breath, all of that kind of stuff associated. But not like shortness of breath walking up those stairs to get into the building. You're like, oh my God, I'm 85. Yeah, that kind of shortness of breath. Not like in Redwood when you get up to the third floor, because literally everybody is winded by the time they get up to the third floor. They are. I always think, I'm totally getting old. And then I watch you 20-year-olds and I'm like, I got you, man. You're just as bad as me. I know, everyone's like, okay. Now, let me tell you where we used to get EPO. It's not a very pretty story. Okay. So the place that we used to get EPO, it wasn't, so if we needed to use EPO clinically, and sometimes we do, for instance, if someone has leukemia and we have to destroy their bone marrow, it's going to make it really difficult for them to recover if they don't have any oxygen being carried around. So one of the treatments would be to add EPO back to their system. Does that make sense? And there are a couple other reasons that you can shoot them up with EPO.
Destroying their bone marrow, then.
But we also do a bone marrow transplant, and then we have to make a new bone marrow make. Does that make sense? Okay, got it? Okay, so I didn't get that far. So destroy the bone marrow, they're screwed. We give them a bone marrow transplant, and then we need them to make red blood cells, and we can increase that with EPO, erythropoietin. So where we used to get it, it wasn't readily available. Where we used to get it was not a very nice story, not a very good human rights, social story. Where we used to get it was we got it from countries that, like really moist equatorial countries that had a lot of malaria. Because here's what malaria does. Malaria is an intracellular red blood cell pathogen. What it does is it causes hemolysis in red blood cells. So it infects the red blood cells, causes hemolysis. And so, of course, they go really hypoxic and they're exhausted all the time, right? And so when malaria is quite often chronic, and they are going to make boatloads of EPO, so much that they end up peeing out half of it. We used to buy the urine from countries with high levels of malaria. I'm really hoping, but I'm cynical at this point, that we also gave them lots and lots of medication for malaria, but I'm no longer sensitive as I used to be.
Get it
I know. We're just like, oh my gosh, this is so uncomfortable. So that's where we used to get it. Thank goodness. Now, we did not have a change of heart, but we did figure out how to genetically engineer it in the lab. This is not where we get it anymore. We don't buy urine from other countries anymore. We make it in the lab. Now it's more readily available, and of course, it's more likely to be abused, right? So who would like to have a really good oxygen carrying capacity? Athletes, especially competitive athletes. So how do you make sure that you versus the other cyclists, because it's really, really common in cycling, are going to have greater oxygen carrying capacity? You shoot yourself up with EPO, or you do it the sort of more natural way, which is give your own blood, spit it down, keep your red blood cells, wait 2 to 3 weeks until you've regenerated your red blood cell volume, and then shoot yourself back out with your own red blood cells. Okay? So how the hell did they figure out whether you got that naturally because you're like a super duper athlete or unnaturally? Now they don't care anymore. They just say this is the maximum number of red blood cells you can have, and I don't care how you got the rest, but you're not going to compete if you got any more than that. And it's a really high number because they're athletes, right? And it's going to be higher for men than it's for women for reasons I'll describe in just a minute. So is it dangerous to do this blood doping? Yeah, it is. Because if you inject double the volume or even 1.5 times the volume of red blood cells, think about the viscosity of your blood. Yeah, it can cause erythrocytosis, which can cause heart attacks and strokes. You just can't pump it adequately. And people who are perfectly healthy athletes have died because of blood doping in that situation. Okay, so Procrit is, by the way, a medication. It's one of the first ones that I knew about. I'm sure there's way more now, which was a medication that was basically EPO, usually used for cancer patients, but not always. Okay, so testosterone also has this little effect. Testosterone plus EPO means greater erythropoiesis than EPO alone. So testosterone increases red blood cell synthesis in the presence of EPO. So causes the kidneys to release EPO. So the red blood cell count in a male versus female, not only do males have more blood on average, it's just based on body size though, but in a drop of male blood versus a drop of female blood, there's also much greater oxygen carrying capacity because per cubic millimeter or a milliliter, a big drop, right? Males have almost 1 million more red blood cells per drop than females have. So way greater oxygen carrying capacity. On average, males have 5.4 million red blood cells per mil, and women have 4.5 on average. Now, of course, Serena Williams would have more than all of you dudes, right? But if you're looking at the bell curve, the male bell curve is a little further that way than the female bell curve is. Is that good? Okay, so for that reason, among other reasons, and with elite cardiovascular athletes, do males and females usually compete against one another? No, they don't, right? Males have greater muscle mass and greater oxygen carrying capacity than females do. Okay, so because of that, by the way, males end up with more hemoglobin simply because they've got more red blood cells than females do. So let's talk about hemoglobin, our friend. Here's our friend hemoglobin. All right, so with hemoglobin, I forgot to tell you something. I'll come back to it in a second. With hemoglobin, it needs to be made, of course, in the red bone marrow, but it needs to be made before the red blood cell has ejected its nucleus. Do you guys know that mature red blood cells don't have a nucleus? That's why they're biconcave discs. Does that make sense to you guys? Okay, so the process of red blood cell production is going to involve, of course, erythropoiesis, but also it's going to involve the protein synthesis of the hemoglobin. You have to have a nucleus to do protein synthesis. And I know I'm torturing you with 1610, but it goes DNA, RNA, protein. You heard that one time before? DNA, RNA, protein. So if you ain't got no DNA, you ain't making no hemoglobin. So it is a red iron-containing pigment and a protein attached to it. So if we look at hemoglobin, This is the heme portion, and this is the globin portion. So keep that in mind because one of them will carry one thing. Heme, yes, iron-containing portion. Globin, globby protein. Got it? Okay, so keep that in mind. All right, so we know this about hemoglobin. It carries oxygen, and when oxygen binds to iron, rusts, right, makes it redder. So your hemoglobin is redder when oxygen is bound to it than when it's not, right? So you guys know that in systemic arterial blood, the blood is bright red. In systemic venous blood, it's dull red. And if you go to pulmonary circulation, in systemic and pulmonary venous blood, it's bright red. In pulmonary arterial blood, it's dull red, right? Okay, so one thing that I forgot to tell you, which is kind of interesting before we get into the total function, I forgot to tell you what you do with old red blood cells. Okay, I didn't tell you guys this, did I? Okay, so when we're going through this whole red blood cell thing, you make 2 to 3 million per minute, per second, but they get the crap beat out of them as they're going through capillaries. Capillaries are sometimes hardly bigger than the actual red blood cell, and sometimes they have really acute turns in them, so they really like get physically beat up. So what happens is they last on average 120 days, but you're breaking them down and making new ones all the time. So here is how you know to make new ones. Every single time your red blood cells go through the liver and the spleen less so, but the liver mostly. Do you remember learning that the liver has sinusoid, these big, wide, porous capillaries in it? Yes? Okay. And the sinusoid capillaries have these macrophages associated with them, these big macrophages. So imagine that blood is going by, and I'm sitting on top of the sinusoid capillary, and I'm a big old bulky macrophage, right? Now, usually you think of the macrophage as looking for pathogens, but in addition, they're giving each red blood cell a check every time it goes through. And if it looks like it has been rode hard and put up wet, you know that Texasism? Well, I rode horses growing up, I know, right? If it looks like it does not have another round in it, then the macrophage actually recycles it. So it reaches down, it gloms it up, it breaks down the amino acids, dumps them into the bloodstream for use for whatever you want to use amino acids for, out of anabolic, catabolic processes, whatever. And then it takes the iron and dumps it into the bloodstream, and you're going to send the iron back to where?
Marrow.
Yeah, you're going to send it back to the bone marrow to reuse it, right? But there's a little bit that you can't recycle, and that little bit that you can't recycle is nice and grossly pigmented. It's kind of a greeny yellow color, and what you do is you end up dumping it in as bilirubin into bile, which gives bile mostly the color that it has, and you use bile to dump into the digestive tract to help, among other things, with lipid digestion, right? And then the bile ends up in your poop, which is why your poop is that unpleasant color. It's mostly broken down red blood cells. You know your kids would like that little piece of trivia, don't you? They totally would. And your pee also is that color mostly because of broken down red blood cells. Okay, so that's the sort of recycling of hemoglobin. So we are able to recycle the vast majority of hemoglobin. So you see bile right there, bilirubin. All right, so let's go into what hemoglobin does. And you guys already know what hemoglobin does, and we're going to look at this in more detail in the next 2 sets of notes. But basically, hemoglobin is a gas ferry. Now, I know that we are used to thinking of it as carrying oxygen, but I'm going to tell you that it also carries carbon dioxide. So here's what it does. It's a ferry, right? And it's got 4 seats for oxygen. Yes? Okay. And these seats are only used for oxygen under normal circumstances. I'll give you an exception in just a second. So these seats are only used for oxygen. It can carry 4 at a time. So generally what happens, guys, is as the hemoglobin goes through your pulmonary capillary beds, you pick up 4 oxygens, right? That makes sense? And then you're going to send it to your systemic capillary beds, and they're all going to get off the bus, yes? Okay. But at the same time, what you are going to do is you also have the capacity to carry on hemoglobin. You can carry carbon dioxide, but it doesn't use the same seats, and this is important. So it won't outcompete oxygen. It is carried not on the heme portion, but it's carried on the globin portion, right? So does it make conceptual sense to you that as you're going through a systemic capillary bed, you could actually pick up some carbon dioxide before you drop off the oxygen? Yeah, so you can actually carry those at the same time. You don't for a long period of time because as you're going through the capillary bed, you're generally going to drop off most of the oxygen, right? So when it has oxygen bound to it, it's called oxyhemoglobin. It tends to be bright red, right? When it has carbon dioxide bound to it, it's called carbaminohemoglobin. Now, let me just briefly talk to you guys about carbon monoxide poisoning, even though we actually do it in the respiratory system too. What is the issue with carbon monoxide versus carbon dioxide? The problem with carbon monoxide, which of course you get exposed to by burning fossil fuels or anything organic, right? And it's this colorless, odorless gas, and you guys have heard of carbon monoxide poisoning, right? So what's the issue with carbon monoxide poisoning? If it's in the air, the problem is the little bastard takes up oxygen seats and will not give it up. So every single time you breathe it in, it has a greater affinity for the heme than, just had to make sure it wasn't my kid, has a greater affinity for the heme group than does oxygen, yes? And every single time it goes through your lungs, you pick up more of it and it continues to outcompete oxygen and it doesn't let go of the seat. Does that make sense? So you end up basically poisoning yourself. You're poisoning and depriving all of your tissues of oxygen by breathing this stuff in. Okay, and it's really hard to get it to give up the seat. Sometimes they have to put you on complete oxygen, like 100% oxygen for a long time before eventually that will go. Okay, so that's carbon monoxide poisoning. It's in the next set of notes, but we already talked about it. Okay, so basically what we are going to do is the vast majority of oxygen is going to be carried on the heme group. But only a small proportion, about 20% of the carbon dioxide is carried this way. Most of the carbon dioxide is actually carried in the plasma, and most of it that's in the plasma is not even carried as CO2. It's carried as, oh, you've heard this chemical, yeah, bicarbonate ion. It's carried as the bicarbonate ion. So that means that you're going to have to learn that damn equation again. But you didn't forget it, did you? CO2 plus H2O yields H2CO3, which dissociates to HCO3 minus and H plus. Okay, so we will do this, and I'll show you this mechanism when we get to the respiratory system. Okay, so we're going to do that in details. Oh, that's where it is. So destruction of red blood cells, we already said that they last about 120 days. When you destroy them, it's called hemolysis. And we talked about bile and poop. Okay, so clinical connections before we leave red blood cells. Anemia is a really general term that means reduced oxygen carrying capacity. Anything that reduces your oxygen carrying capacity would be considered an anemia. Now, you can have nutritional anemias, you can have anemias that are problems with your bone marrow, anemias that are problems with the red blood cells themselves, anemias that are problems with the shape of the hemoglobin, but generally it's anemia. That makes sense? And symptoms of all of the anemias are fatigue, both mental and physical. You got no oxygen, you got no cellular respiration. Shortness of breath, sometimes pale, right? Sometimes chilly. So let's talk about the kinds of anemias that exist. Hemorrhagic anemia would be the anemia that I had and Charles had and didn't pay attention to right after we gave blood, or right after you hemorrhaged, right? So that would be hemorrhagic anemia. And that is like acute. Generally, you're gonna like regenerate that. Hemolytic anemia would be the anemia that you would have after a mismatched blood transfusion or the anemia that a person with malaria would have, right? Because you're doing hemolysis and busting those things. Is that okay? Aplastic anemia, the problem is not here, the problem is here. What if you take in a chemical either accidentally or on purpose that completely trashes your bone marrow? Then you are going to have an anemia because there's something wrong with your bone marrow. Now, sometimes you do that on purpose, in which you are doing, for instance, chemotherapy because of leukemia, or sometimes you live near Chernobyl and it happens accidentally, right? Either way, if you destroy the bone marrow, not only do you have anemia, but you also have a depressed immune system and problems clotting, right? Because you destroy the capacity to make all of the blood cells, not just the red blood cells. So that's called aplastic anemia. That's usually chemical related. Nutritional anemia is not at all uncommon in women who are menstruating. If you do not pay attention to your iron content and you are menstruating, you can have really big drops of iron that give you what's called nutritional anemia, right? Like, I just lost some vascular tissue and I don't have enough iron to make the replacement, right? So this is not at all uncommon in women. insufficient amounts of almost anything, but quite often iron is the sort of rate limiting one. Folic acid, vitamin B12, protein. So that's called nutritional anemia. Renal anemia. Does it make sense to you now that if you trash your kidneys, you're going to have oxygen problems? Yes? Because if you actually have renal anemia, like let's say we've got a person who has kidney failure because of type 2 diabetes, in addition to maybe being on dialysis, they might also have hypoxia because their kidneys are trashed. Does that make sense? So they might need treatment for that as well. So that's called renal anemia. Go ahead.
So is that because they're not producing erythropoietin?
Exactly. When your kidneys start to completely fail. And usually kidney failure does not immediately affect hormone function, but as they start to really fail, they lose all their functionality in the late stage of kidney failure. Does that make sense? Okay, and then go ahead.
Does that get fixed with the transplant?
It would be fixed with a transplant if the transplant was not rejected. However, a type 2 diabetic who's not regulating their sugar properly cannot be on the transplant list. So you are not eligible for a transplant if you have untreated, unmanaged diabetes that got you on the transplant list, which seems heartless, but it also seems reasonable. Right? I don't know. Pernicious anemia. Pernicious, that word sounds like an old person. Pernicious, and it's an old people anemia. Here's the deal with vitamin B12. You need vitamin B12, we said, in order to make red blood cells. And you need this stuff called intrinsic factor, which is made in the lining of the gut, and you lose the ability to make adequate levels of it as you age. So you tend to get this pernicious anemia in which they're really exhausted, don't have enough red blood cells. But would giving them vitamin B12 supplements help? No, because they still can't absorb it. So what would you have to do? Yeah, you'd have to give vitamin B12 injections, which are not at all uncommon with elderly people. Does that make sense? Okay, so hereditary anemias. I know that you guys have heard of this one before, but I just want to make sure that I briefly do it. Sickle cell disease used to be called sickle cell anemia, and I know you guys have heard the sickle cell story before. Have you heard the sickle cell story? Okay, so I'm just going to do it briefly then. So sickle cell anemia or sickle cell disease is, no, not that one. Okay. All right, so it's a genetic disorder. It's autosomal recessive, which means in order to have it, you usually have to have 2 of the same mutation. Here's what it does. It causes a change in a single amino acid in one of the 2 chains of hemoglobin. I know you got 4, but 2 of them are identical to each other, okay? And the deal with that is that the hemoglobin folds differently, and therefore what happens is the red blood cell, which is primarily just a bag full of hemoglobin. If you get a red blood cell in hypoxic conditions, it will sickle. Okay, now this can kill you dead, and does quite often. Well, usually, historically speaking, they didn't live past adolescence. They're living longer now. So it gets stuck in your capillaries, can cause heart attacks, strokes, pulmonary embolisms, and it's super duper painful. But it's still really, really common, which is counterintuitive. If it killed people off in adolescence, how the hell did it become so common? And that weird little thing is that this genetic disorder, which is most common in people of African descent, but also some portions of Asia, has a little weird overlap with malaria. And here's the overlap. Malaria likes to infect red blood cells, and it can kill you, yes? But it only likes normal red blood cells. It does not like sickled or partially sickled red blood cells, okay? So malaria can kill you, and you're not protected against it if you do not have a sickle cell gene, right? If you get double sickle cell genes, you get this and die. But who has an advantage to both? A carrier to sickle cell disease. Because a carrier does not generally sickle, right? But their cells are slightly weird shaped, and malaria doesn't like to infect them. So historically speaking, who was more likely to survive? Yes, sickle cell carriers. So we end up with a situation like we have now, which is 60,000 to 80,000 people of African descent still have sickle cell. And it continues to be propagated because we haven't eradicated malaria yet either. Go ahead.
When the macrophages in the liver, would they destroy them?
Yeah, they do break these down really dramatically. Yeah, they're like, that's weird looking, and then they continue to have more hypoxic conditions. Yeah. So does that make sense to you guys? So this is a weird disorder in which there's a link to malaria that actually keeps it in the population. Okay. Other anemias, if you are super duper lazy, you don't need very many red blood cells, and you might make very many. Also, hypothyroidism will cause anemia as well because your thyroid isn't supporting it. Okay, so, hemoglobin. Okay, so now we are going to move to platelets and hemostasis. You ready? Okay, so first thing is you need to understand that the word hemostasis is different than homeostasis, although it does lead to homeostasis on a good day. Hemostasis implies, hemo implies what? Blood, and stasis implies stability. This is broadly the cessation of bleeding after an injury, and it includes clotting, but it's not limited to clotting. Hemostasis is a 5-stage process to stop you losing blood. You with me so far? Okay, so hemostasis. So what I want to do before we get into details is I want to go over the big picture of hemostasis, and then I'm going to go back and add the details. You ready? Okay, so I want you to imagine that I've just either given blood or had an IV or something, and what I've done is I have punctured, let's say, a vein, because arteries are a little harder to clot because they're super high pressure. I mean, you can clot them, but they're a little harder. So let's say I've punctured a vein, right? And so what I have is a hole in a vein. I've basically got a hose, and it's got a hole in it, right? So there are 5 stages to actually stopping you from losing fluid. And the first stage, you may or may not have ever heard of, but it should make sense. If you got a hole in the hose and you can't turn off the fluid, what do? You kink the hose, exactly, right? So the first stage is vasoconstriction, upstream or downstream of the wound? Upstream of the wound, exactly. So you kink the hose upstream of the wound. Good? Okay, that makes sense? So just keep that in mind, and we'll go back and look at how that occurs in just a minute. So we've got vasoconstriction. Then after vasoconstriction, what happens is you start to attract these platelets, and it's called a platelet plug formation. So I want to show you something so that you don't get confused. A platelet plug is platelets congregating at the site of the wound. And I'll tell you why they do that in just a second. But first thing is I want you to understand that it's platelets congregating. That is not a clot. Let me show you what a clot looks like. A clot is a whole bunch of sticky spider-webby fibers that actually are generated and are insoluble. So a platelet plug precedes a clot quite often, but it's not the same as a clot. Okay, so platelets are congregating. I always think of platelets like teenagers. If you do not supervise them properly, they are going to congregate. Somebody's going to get pregnant. They're going to snort something. They're going to drink all your booze, right? That's platelets. These are not being properly supervised. Somebody told them that there was a basement with a keg in it, and they're just hanging out, right? Okay, so that can lead to a clot, and the clot is the next phase. The next phase is called the coagulation phase. Coagulation means I'm forming all these sticky, insoluble spider-webby proteins that are going to plug the hole while I'm repairing the hole. Does that make sense? So this is a clot. This picture is a clot, okay? Now, coagulation phase. And then after that, the next is called the clot retraction phase. And what happens after you coagulate is these fibers will actually yank on the edges of the damaged vessel and pull them closer together to make healing easier. Does that make sense? That's called clot retraction. And then the last phase is when you are completely done with the whole damn thing and you need to dissolve the clot so that you can have restoring normal blood flow. That last stage is called fibrinolysis because these spider-webby proteins are fibrin. Okay, so let's go through the 5 stages of hemostasis again. Ready? What's the first stage? Vasoconstriction. The second stage? Platelet plug formation. The third stage? Coagulation. Fourth? Clot retraction. Fifth? Fibrinolysis. Only one of those stages is actually complex. It's only coagulation that's complex. The rest of them are relatively simple. Okay? Everybody okay? So now we are going to go back and add detail. Give me just a second to be rude because my kid is, okay, there we go. Like if he needs me, I'm out, sorry. Okay, so now let's go back and look at the first stage. So the first stage is called a vascular spasm or vasoconstriction. And this is basically smooth muscle in the wall of the vessel constricting upstream of the wound. And this happens in every vessel except a capillary. Why can it not happen in a capillary? There's no smooth muscle in a capillary, so they can't do the vasoconstriction phase. Okay, so the cause has a question mark on it because they think it's caused by paracrine agents, but they think it also might be a nervous system response to damage. They don't know the second part yet. So one of the things that we do know is that upon damaged, platelets will start at the area and they'll release some things that will cause vasoconstriction upstream. They're paracrine agents. Now, you don't have to memorize these paracrine agents, but I'll just tell you that they're weird. Like ADP is used as a paracrine agent here. Thromboxane A2, anytime you see the prefix thrombo, it means clot. And serotonin is actually used as a paracrine agent here. And so those are released and they actually cause vasoconstriction in the area. Are released primarily by platelets, and this lasts for a long time. This vasoconstriction lasts for up to 30 minutes because you do not want to unkink the hose until you've done what? Until you've repaired it. Exactly. You need to stop the fluid, and you also need to repair the hose, right? So this lasts for up to 30 minutes. Okay. All right. So the next phase is interesting. So the platelet phase or platelet plug formation is next. Okay. So here's what happens with platelet plugs. When platelets encounter something that they have not seen before, they will start to send a group text to all their little platelet friends. So what happens here is like somebody's parents are not there and they got a keg in their basement, right? And then they start sending out a group text. In this particular instance, how would a platelet know that something is weird? Well, when a platelet encounters a collagen fiber, that means that the vessel wall is probably damaged because the endothelium of vessels, does it have any collagen in it? No. So what you're seeing is the connective tissue deep to the epithelial tissue is now exposed. Yes? And that is usually the trigger for platelets to start to congregate. So how do they start to congregate? Well, first thing is, as soon as they get activated by coming into contact with that collagen fiber, they become a little bit polar. And a little bit polar means that they're slightly positive on one end, slightly negative on the other end. That alone will attract their friends as they go by, right? A platelet isn't a cell, it's a cell fragment. It becomes a little bit polar. And then in addition, they'll start to release a whole bunch of paracrine agents that are all pro-platelet congregation. And it's sending out a group text, right? It's like, hey, there's a party, come on. And then they start a platelet party. And here's my platelet party. This is my favorite cartoon. You ready? Blood loss, platelet party. No serious business. There's platelet. Party! So the platelet party starts whether you want it to or not. And sometimes it can even start without collagen. That's one of the reasons that platelets need to be supervised, because if you get one of them and you start sending out a group text, then a party's going to start and the basement ain't even empty. That's the problem with a situation like mine with atrial fibrillation. So why would I start an inappropriate clot when there was no damage? Because what do my platelets do if they don't get squished out of my atria? Well, they just start thinking, nobody actually told me there was a place to have a party, but since we're all here, and then they start attracting one another. Does that make sense?
Yeah.
So, what?
They're slightly polar then.
They start to release paracrine agents as soon as they get close to one another. So you always want to keep the platelets moving along, right? As soon as they get close to one another. So you don't have to have collagen. That's the easiest way to start it, and that's the way that it's sort of meant to. But if I had atherosclerosis and my blood vessels were really lumpy, and then they just get caught next to one another, that'll start it too. Or if you have atrial fibrillation and they're just hanging out, then they're going to start a party, right? They're like 5 teenagers. You're going to have a party. Okay, so this platelet congregation or platelet aggregation is going to start a platelet plug. Now, let me just tell you a couple little interesting things. There are some other paracrine agents that are involved. All kinds of paracrine agents are released that cause positive feedback loops. I don't usually ask you these by name. I do usually ask you a bunch of the factors in coagulation when we get to that, though. Now, just because it's kind of curious. Epinephrine and serotonin, ADP, prostaglandins, which we learned about way at the beginning of the semester, thromboxane A2. These are all released to cause a little positive feedback loop that says, come on over, come on over. And in addition, there's this weird one called von Willebrand factor. And von Willebrand factor is a paracrine factor that is secreted by platelets and also damaged cells. And it's one of the positive feedback loops. And this one's historically important because von Willebrand hemophilia was relatively common in the European royal families. It's a genetic disorder in which you do not make one of these paracrine agents. Did you guys know that the European royal family had hemophilia running rampant through it? And so what happened is, like, somebody had a mutation, but then they also married their first cousin over and over again to keep the royal line together. So you ended up with a homozygous recessive situation over and over again. And a bunch of the heirs to thrones in different European countries died, right? Especially because this one is more likely to be manifested in males. Okay, so these are platelet plug. So is a platelet plug the same as a clot? No, but it's a temporary placeholder for where a clot could come, right? So this takes about a minute to form, a platelet plug. And sometimes the platelet plug is big enough to fill like a small hole. You don't need to do coagulation. It usually does it anyway. But usually what you're going to have to do is you're going to have to do coagulation after, because you will see that coagulation actually helps with healing as well. So the platelet plug formation starts a little positive feedback loop. But here's an interesting thing. Why do you not continue to go willy-nilly platelet plug formation both ways? Like, why does that not happen if it's doing a positive feedback loop? Well, because interestingly, at the site of damage, platelets are going to attract one another and continue this positive feedback loop. But all of these undamaged tissues are going to be going, move along, kids, move along, right? They're going to be releasing things that cause the platelets to not stick. So do you see that although at the site of injury, you are going to be doing positive feedback, at the edges, you are going to be limiting it. Does that make sense? By negative feedback. That makes sense? So it keeps the platelet plug from just going willy-nilly. And by the way, you are always releasing these from healthy tissues. In our healthy tissues, why do we not form platelets more frequently? Because our healthy tissues are generally saying move along. Does that make sense? Okay. All right. So these are just some of the ones that you actually use. All right. So aspirin therapy. You guys have probably heard that people who have a predisposition to clotting sometimes do a daily dose of aspirin therapy. The aspirin therapy is related to this, because what aspirin therapy does is takes out prostaglandins, just one of the factors involved in the positive feedback loop, and makes it less likely that you are going to form an inappropriate platelet plug and therefore less likely to clot. Does that make sense? So interestingly, they always call it a blood thinner, but it doesn't thin the viscosity of your blood, so I don't like that term. It's also actually not an anticoagulant because we haven't even gotten to the coagulation phase yet. It's antiplatelet plug formation, which makes it less likely that you'll form coagulation or clot. Got it? Okay, so I am supposed to be taking that, 80 milligrams of aspirin per day to prevent inappropriate clotting. Why do I not? Well, I'm not a very regular person, first of all. And second, every single time I run into the freaking coffee table, if I'm on 80 milligrams of aspirin per day, I get like a whole leg full of bruise. Because not only does it prevent appropriate platelet plug and clot formation, it also, or prevent inappropriate, it also prevents appropriate, right? So I bleed a little bit more every single time I have a bruise. Okay? All right. So now this is where it gets a little challenging, so make sure that you're paying attention if you were not before. Coagulation phase is the only hard part of this entire thing. And it's one of those things that seems really complicated when you first talk about it, but then it will clarify. You ready? Okay, so here's the general information. Because platelets like to congregate, if you let them sit next to one another, they will start to congregate and then they will start to release things that form clots. Okay, so if you just draw blood and stick a drop of blood on the table and leave it there for a minute, it will eventually gel. And when it gels, what happens is you have actually started to form this insoluble sort of mesh of proteins, which, by the way, is what gel is, a little insoluble mesh of proteins. And then everything, have you ever done that? Have you ever seen blood just pool for a second? What happens is it will gel in the middle and then it will push out some clear yellow fluid around the edge. That clear yellow fluid is called serum. And it's basically blood plasma minus all the clotting proteins because those stayed. Or have you ever had a road rash? You ever had a road rash? If you have a road rash, what you're doing is you're clotting on the surface. And when it starts to pull together, it will push out that clear yellow fluid, not inflammatory fluid, not pus, because that is opaque, right? But the clear yellow fluid that you get pushed out during road rash. Go ahead.
How come that doesn't happen when you donate blood?
Because, oh, that's a great question. Hold on to that question. I'll explain it in just a second. Okay, so the gel is basically this. The gel is this clot with the sticky spiderwebby proteins called fibrin, and anything that was going by got caught in the spiderweb. So were these red blood cells actually part of forming the clot? No, but they got caught as they were going by, right? Okay, this is a platelet right here. This is a platelet right here. Here's another sort of close-up view of red blood cells caught in this spiderwebby protein. Everybody okay with that? Okay, so the gel is the clot. All right, so what clotting is, guys, is a domino reaction, a cascade effect. Do you remember how we said, okay, so what was, you said it was similar to what?
The renin-angiotensin.
Okay, yeah, so renin-angiotensin aldosterone system. Do you remember how we activated one thing, then that thing activated another one, then that thing activated another one? Blood clotting is basically a big, long cascade of those, of a whole bunch of things that are always in the bloodstream, but they're inactive. And then if you get one started, it'll activate another one and activate another one. So I'm going to show you this, but don't freak out when I show you, because I will simplify it for you. It looks terrible when I show it to you the first time. Okay, so this is what blood clotting is. Okay, I can simplify it for you immediately, first off, because what's going to happen is this is inactive 12, but when you activate it, it's 12A. This is inactive 11. When you activate it, it's 11A. Okay, so what's going to happen is once you get one of these, it's going to activate the other one and the other one and the other one and the other one until ultimately down here what you get is the clot, it's the spider web. Okay, now here's what you do not have to do. You do not have to memorize all of these, but you do have to memorize a few different branch points, like a few important parts of the story. Is that okay? Okay, so this is called a coagulation cascade. It's like a domino effect. So let me ask you, conceptually, if I tell you that all of these that are sort of in the lighter color are always around, but inactive in the bloodstream, why would you rather do it that way than just make all of these when you needed a clot? Yeah, so this is fast, right? So what I have is all of these inactive things, and if I get the first one started, it'll activate the second one, activate the third one, activate. Does that make sense? So what happens is all of these clotting proteins are present but inactive in the bloodstream, but they need to be able to be on at a moment's notice. And that's what this does. It's going to actually be able to turn all of this on at a moment's notice. Okay, so do you have a question?
You want it to be fast. Why is there so many steps?
Mm,
Would it be faster if there's just one thing to activate?
that's a good question. I don't know the answer to that. I'll think about it. So, what?
Is it like the more powerful, like iron?
I don't know. Well, there's two different ways to do it, but once you do it, you've got it this way. There are a bunch of different factors that are needed. I don't know. Maybe it just evolved that way. I've never thought about that before. Okay, so let me tell you another important thing. Most of these proteins that are there in an inactive form are made by the liver. Ding, ding. So if you got liver problems, you got clotting problems. Okay, so the vast majority of them, not all of them, are made by the liver. And this domino effect, so all of these that you see in the lighter color are present all the time, but they won't actually get activated unless they have a trigger. So we're going to start talking about the triggers in just a second. You ready? Okay. Ultimately, this is the goal right here, which is this fibrin, the spider webby stuff. All right. So each clotting factor, by the way, I teach this slightly different than some phys professors teach it. I do not make you memorize the numbers, in part because I suck at memorizing the numbers. But the important branch points you will need to know, and you will need to know some other things. But let me just tell you for future reference. Generally, these are Roman numerals. If you do not know Roman numerals up to 12, you probably shouldn't have passed anatomy, right? Because there's 12 cranial nerves, and people come up on that test all the time, and they go, what is IX? And it's like, I should have studied is what IX is. So I won't make you memorize very many of the numbers, but 12 and 12A are related to one another. That's activated 12. And they were numbered, unfortunately, this is super annoying. They were not numbered based on their position in the coagulation cascade. They were numbered based on when they were found. So you get like 8 and 12 and whatever. It's like completely annoying. What? Yeah, so they were numbered in the order that they were found. I know this factor is involved in blood clotting. I'm going to name it this. I found this one. And then they found another one. And then they found another one. That's part of the reason that a person like me who has an atrocious time memorizing numbers does not make you memorize the numbers. But you need to be able to understand it, right? You're like, okay, I see 9 or 9A, right? I got them. Okay, there are a couple numbers that'll make you memorize. Okay, so clotting is slower than platelet plug formation. Clotting takes on average 3 to 6 minutes. How long did platelet plug formation take? Yeah, 1 minute. And here's something else that's important. It requires calcium at multiple different points. Haven't I told you all semester long that you use calcium for blood clotting? I have. Okay, so calcium, 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 calcium. Okay, so that's why, among other reasons, that if you are deficient in calcium, you will, for instance, take it from your bones. Because if we had to choose between bone mass and blood clotting at any given moment, you would probably choose blood clotting, right? So calcium is needed for this process to be functional. Okay, so here's what we're going to do. We're going to look at the stages. So the first stage, I'm going to start at the bottom, and then we'll work our way back up. So please look that at right here. Once you get right here, do you see this factor 10A right here? Once you get right here, you get a clot, don't you? Okay, so here's what I want to do. I'm going to start here, and then I'll go back and pick up how you get there. So the first stage will be getting to factor 10A, which is called prothrombinase. So let me show you how prothrombinase is involved in here. Okay, so what we have is prothrombin. Okay, watch. Prothrombin gets converted to thrombin by prothrombinase, which is one of the few that I will make you memorize by Roman numeral, which is factor 10A. Okay? So please look at the names because it's not all rote memorization. What does pro mean? Before. Prothrombin gets converted to thrombin by prothrombinase. You okay with that? We still haven't gotten fibrin, though. We almost got there because thrombin converts fibrinogen, as in the genesis or the beginning of, to fibrin. Fibrin gets stabilized, and that's your clot. This is what we were trying to do. Okay? Fibrinogen, as in the genesis or the beginning of, fibrin. Okay, so before we go any further, which of these were always around? Which of these were always around? Fibrinogen. But under normal circumstances, they have no interaction with one another, right? Unless there's actually a trigger of 10A. Okay, we'll come back and figure out how you get 10A in just a second. Everybody okay with that? Okay, so let's go back and figure out there's 2 possible ways to get to 10A. Do you see that there's 2 ways? This pathway to get to 10A or that pathway to get to 10A? Do you see that? Yeah? Okay, so prothrombinase can be formed in 2 different ways. Okay, so let me just do the terminology and then I can have you guys figure out the other stuff. Okay, so there's 2 different pathways to form prothrombinase. There's the extrinsic pathway and the intrinsic pathway. Okay. So one of them, listen to both before you match the terms. One of them is initiated by platelets. The other one is initiated by tissue damage in the wall of the vessel or the tissues around it. So which one will get the name intrinsic pathway? Platelets or tissue? Yeah. So this one is initiated primarily by platelets. Platelets get involved over there too, but they don't start it. So platelets. And this one is initiated by the tissue damage, either the tissue around or the vessel wall itself does the initial. Okay. So now, watch. So there is a lot of detail in each one, but there's one important thing on each one. Let me show you. Okay. So if you initiate it with tissue damage via the extrinsic pathway, you are going to eventually release something called tissue factor 3, TF3. Okay? So tissue damage, extrinsic pathway, tissue factor. Got it? And that will be how you get to factor 10A. But if you initiate it ultimately with platelets, usually by contact with collagen, although they can start it without, then eventually instead of releasing tissue factor, they are going to release platelet factor. Okay? So to clarify, tissue damage causes the release of TF3, tissue factor 3, which will get you to prothrombinase. Yes? And platelets will release PF3 to get you to prothrombinase. Got it? Okay, so let me show you one more time. Ready? Okay, and then I'll do the summary for you and simplify it. So ultimately, your platelets, assuming that they came into contact with collagen or just started misbehaving on their own, will release platelet factor 3, which will get me to prothrombinase. Does that make sense? And these guys will release tissue factor 3, which will get me to prothrombinase. So let's look at the summary, and then I'll show you an image that I'll post for you. Ready? Okay, so when you get here, it doesn't matter how you got there. So from about the time you have prothrombinase to the bottom, this is called not extrinsic or intrinsic, but the common pathway. Okay, so let me show you the summary that will clarify this. Ready? Okay, here's how you read this. Okay, the extrinsic pathway, guys, is activated by tissue damage, yes? And tissue damage releases tissue factor 3. Tissue factor 3 ultimately activates prothrombinase. Prothrombinase converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin gets stabilized to form a clot. Okay? Last one, intrinsic pathway. You ready? The intrinsic pathway is activated usually by the exposed collagen causing your sticky platelets to release platelet factor 3. Yes? Okay? And then the platelets are directly involved in this. And the platelet factor activates prothrombinase. Prothrombinase converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. It gets stabilized to form a clot. Do you see? Okay. Now, for me, you do not have to memorize all of the intermediate steps. Okay? There's a simplified version. And I have this that I will post for you guys today, okay, that has in red all of the parts that I expect you to be able to fill in if I asked you. You ready? Okay. So this is a simplified version of it. Let's read through it. You ready? Okay. Tissue damage would activate which pathway? Extrinsic pathway. There's a lot of other things involved, right? But the extrinsic pathway will ultimately cause the release of tissue factor 3, yeah? And tissue factor 3 will get me to the common pathway, in which you make prothrombinase, which is 10A, right? And prothrombinase converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin, and that's the clot, right? And then the other way, instead of starting with tissue damage, you start with the platelets. Platelets release platelet factor 3. Lots of other stuff goes on that you do not have to know by name. This pathway is called the what? Intrinsic pathway. That's how you get to the common pathway. And then you get factor 10A, which is prothrombinase. And what does it do? Converts prothrombin to thrombin, and thrombin converts fibrinogen to fibrin, and that's the clot. Okay, so I will post this for you. It didn't come from your textbook. It came from another textbook. But I think it's a simplified version that's actually more visual of what's actually happening there. Is that okay with you guys? Okay, so next time, I'm going to take the last 2 minutes, and then I'll... next time, all we have to do is clot retraction and fibrinolysis. But let's acknowledge that even though I am not holding you responsible for every single factor involved in clotting, the clotting is pretty damn complicated. There are at least 13 different factors involved in clotting if you consider the activated one and the inactivated one a different factor, and also if you consider the calcium and all of that kind of stuff. You're only responsible for the factors that are described in your notes. That's it. It requires calcium at multiple points, so even though I'm not having you memorize this, which pathways use calcium? Intrinsic, extrinsic, or common? Yes, all of them use calcium. So if you got a calcium problem, you got a clotting problem. And then the last thing before you guys leave is that the other thing that's necessary for clotting is you have to have prothrombin, yes? Or none of this will work, yes? So do you see that this is a crux point right here? Well, one of the things that you need in order to make prothrombin is vitamin K. Vitamin K is produced primarily by the natural flora in your large intestine, and it's absolutely necessary for normal blood clotting. You guys with me so far? So the last thing I need to say about vitamin K is important, for instance, for pregnant women. Immediately after a baby is born, their large intestine has not been adequately colonized by the normal flora, right? And so they do not have much vitamin K, which means what about their likelihood of, for instance, bleeding out if you poke them? It's pretty highly likely. How did we get around this in most relatively modern countries? We give newborn babies a vitamin K injection. And it's just meant to be protective until it protects them for about 8 days while their gut's getting colonized. And as their gut gets colonized, they start to make their own. Interesting little unintended side effect of the anti-vaccination campaign is that a lot of poorly informed parents think that everything that you inject into anyone is a vaccine, and they are now refusing vitamin K injections for their babies. And we are having preventable baby hemorrhages. Because sometimes you have to poke them, right? Sometimes you gotta poke them to figure out what's going on in there, right? And you don't want them to hemorrhage. So that's the vitamin K injection thing. My OBGYN is a high-risk OBGYN because I had a heart problem, and he told me that he has had more of this in the last 5 years than he had in the last 25 years of people rejecting vitamin K injections. A little information is a dangerous thing. Dangerous thing. Okay, so I will see you guys on Monday, and we're doing respiratory, right? Respiratory next. This will just take a minute to finish.
Just so everybody knows before this goes, there's an active shooter
No.
situation happening.
It would be difficult to be. I mean, there actually has been. There was a famous case in Canada in which they had a homeopathic